So we missed one class last time, but we should have enough time to do what we need to do. So first, please upgrade your single cell gene expression analysis toolbox. You can Google it and find this page in your MATLAB. Please just rerun this quick installation. You should overwrite the previous version and get an update. Well, this depends on how quick your computer is, and it may take 1 or 2 minutes. I made some changes. For example, one of the change I made is based on your feedback. And one of you ask, can we have a metadata? And so we know later and where I get my data in, where's the data source. So I add that function. So now if you download your data from a geo, and the accession number will be associated with your data set. So next time when you forget, you can take a look, and I will show you how. And also, if you merge two data sets, the accession number will be saved as well, and so you can reconstruct your data set later. So just go ahead and bring up your MATLAB and run this code and upgrade to the latest version.
Dr. Kai, we can't hear you online if you're saying something.
Yeah, Alex, you should be able to hear me right now.
Yeah, now
Okay,
we can.
good.
Yes. Yeah.
Great. Yeah, was a battery problem. Okay. So the first paper we are going to try is this paper.
마이 예지 생일 너무너무 축하하고 진짜 예지가 내 인생에 들어와줘서 난 진짜 영광이야. 우리 진짜 평친하고 오래오래 봅시다. 진짜 예지 많이
I wish I know Korean.
사랑해요. I love you.
This question is difficult. Yeah, the question I hope I understand is you just go maybe Google, just search geo. Okay. So on the first page of this paper, you see, okay, they gave this geo repository ID, and I can copy this. Okay, so that's the record. Make sure the citation is right, the same paper, and the cross-validate. So let's see, they have, so if you have something like this, what are you going to do? We haven't read the paper, but if you read the paper, you may know exactly what those 11 samples are. It seems they have a mouse and a rat, 2 different species data. And if I click go into one of the samples, there's nothing here. So what the author did is to synthesize, collect all the samples from different mouse, different treatment, and put together. So they put everything in this folder, this 3. Okay, so if we are going to download the mouse data, so that's a 3 file target we are going to focus on. I will download the data from here. Remember, we can give the toolbox just a session number, but in this case, we won't be able to do this because there's multiple matrix data. There, mouse and the right, 2 different matrix data. So you will confuse the program. So what I'm going to do is here, I will select link to, instead of give this a session number directly. Remember last time I can give the ID, but this time I have to select, I need the link to this mat file. Okay, so now that's the example data. So you need to give the full link, which you can download the file. So what you should do is come back to this page and see, I will right click and copy the link of this file. This, can you see? So because you are going to download the mouse data, so you click this link and copy the link. Right click, don't do the real click, otherwise you don't. So then you put the link to here. You have to do the same thing for the feature which you have a gene name. So I copy this and replace the second, so the genes, and then you can have barcode as well. Copy the link. Okay, I'm ready, and because I found a session number in the publication and come to the record and look at which file I need to download. So I put the 3 files in there to allow the 3 file to be downloaded and load into the program. So that's what the program is doing. So go there, find the link, download the data, and process it, get the data in. Okay, we are done. I'm done with the first example paper. Okay, we're not going to process further. You can do if you can save it later. Okay, we are going to move to next example. See, in for different paper, maybe we have a different situation. As I said, now you should be able to have this view metadata function if you upgrade it to the latest version. If you don't have this option. Then you can take a look, because when you download the data, I already saved the data source into the metadata. So now you should have this available for you, right? So you save the data, the information is still there. Next time you should be able to remember where this data from. You can add additional information. That information will be saved with this data set. So now I'm going to close this. No save. Okay, clean my desktop. We are going to move to the next paper. The second paper is called Single cell RNA identified early diversity of sensory neurons forming by potential intermediates. Published 2020, and let's see where the data is. Okay, at the end, there is data availability section. So here, okay, hope everybody see this. So we have this session code, and I will click in and found this page. And also we check the citation. Yes, that's from the paper we want to get the data from. And then there are 14 samples. Again, I haven't read the paper, but for this case, I just click in one of the samples and see there's nothing under each sample. What that tells you is, again, those individual sample matrix has not been provided by author. Author actually put all the data from different samples into this. So I have to recognize, okay, this is a CSV file, which is text-based, like a table, right? They put everything here because maybe individual sample have two field cells. So they put everything together. So that will be our target. So we are going to download this and process and load into the working environment in MATLAB. So this time is different from a MTX file. So I can do the same thing. I right click the link and copy the link. I come back to my this tool. This time I'm going to select TXT instead of MTX because I know it's in a different format. Well, actually, I can try if I can use a session number directly to download it. So I'm going to try this first using a session number because I have this ID. And I wish the program smart enough to recognize which file is going to download. If not, we have to go back to this, give the link. So let's try a session number. I don't know. So I gave the ID, let the program to decide. Okay, so the program tried to find MTX file because by default, it's supposed to have this MTX. It's not there, so I cannot. I come back and click the file link and copy it. In this case, I select TXT and the GZ file. So this only need one link. I put in there. download the file, unzip it, and put in the temporary file and read that information in. That's all the program is doing right now. Then we should have this cells get in the system.
I'm getting an error that says the file format is not recognized.
Oh, really?
Yeah.
For this particular example?
Yes.
Oh. So I'm doing this, but here I can read it. So I don't know. Are you using the latest version? Can you check the updates? See, okay.
Yes. Well,
Did you copy this file, the last one?
yeah, I tried using the FTP link and the HTTP link because I was having issues with the FTP link on the previous example.
You should be able to get that as well. The HTTP link should also work.
Yeah, it gave me the same error and said it wasn't supported.
So we have an inconsistent. I didn't receive that error message. Anyone
Right.
else had a success story or? Oh, well. So you see, I'm doing exactly the same thing, and the link. I cannot figure out what should go wrong.
Yeah, I just checked mine for updates. I'm on 22.2.9.
Yeah, that's yesterday. Yes, that's a latest version. Again, I don't know. I can't imagine what should go wrong. But that's basically that's a command line. That's unzip this and load that. Yeah, I need more information before I can decide. I can figure out what's going wrong with it.
I'm on 2020A, maybe that's one.
It should okay. Yeah, so I can give it a try for another link, the HTTP link. I will copy this and do that again. So I select text file based input. Oh, yeah. Could be the problem with, can you use FTP link instead?
I tried both of them and I got the same error for both. I can try it again, but yeah, I got the same error.
So on my computer, the FTP link works.
Huh.
Worked.
Yeah, that's actually working for me now.
Okay. So Alex, I'm going to copy this link in the chat and see whether we have a different link copy.
Okay.
So I send it in the chat. You can see. Okay, if you
Yeah.
use this, you should be able to get it. For the HT, copy the link. Yeah, so on my computer right now, FTP link worked. HTTP has some problem with the coding of the HTML. So I may be able to fix it later. Okay.
Yeah, I can just follow along for now. But yeah.
Okay. So the idea is you have to look at the database record and then figure out whether you are going to download MTX file or TXT based matrix file. Okay. So let's move to the third example. So this is a 2021 paper, a subpopulation of swan cell-like cells with nerve regeneration signature identified through single-cell RNA. Okay, I'm going to do the same thing. See, I will search geo. Okay, this tricky part. So it mentioned RNA-seq data used in this study was deposited into here. We have to be careful because this may be not a single cell data because it says RNA-seq data. It could be the traditional matrix. So let me see. Okay, that's the paper. Okay, so when I see this, I can make sure I know immediately this is single cell data because there's only single cell data for now using this MTX files. So there are two options for this, I believe. One is to just use a session number and give to the program. You should have that. And for the individual samples, you don't have data, so everything is here. So in this case, I'm going to just use geo accession number, and then because there are MTX file in there, and we should be able to. The program should be able to help you to parse the HTML code and extract the name and the link for those files. So you don't have to do it copy manually. If you look at the command line window and there are feedback from the program, right now this matrix file has been downloaded and unzipped. So you see it looks the program has been correctly recognized the file names and then doing its job right now. we should be able to get the cells in just a couple minutes. So after this, I'm going to give you 10 minutes for yourself to work out the next two papers without my leading. Okay, see whether you can make it, or in the middle you have any problems. Now it's 10:38. So I will give you 10 minutes for you to process the next two papers yourself. Yeah, Eric, no problem. Okay. Yeah, so the data I'm processing right now looks like a large matrix. On my computer, it already about 5 minutes already. It hasn't been downloaded and processed. It's a huge data set. I don't know how about your computer, and mine is older, I believe. You're still working on that? No. Well, don't know how long it will take eventually. So I will use this time to process and look at the third, the fourth, and the fifth papers.
My thing just finished and it said there was an error setting up property X of class for that data set.
Okay.
Yeah, it says there was a requested 426 gigabyte array that exceeded the maximum size preference of 4 gigabytes.
So it's just run out of the memory of your computer.
Yeah, I guess.
Okay.
Oh, it requested 400 gigabytes, which is
Okay.
quite a bit.
Yeah. Okay. I really should leave this for your homework instead of doing this in class. I don't know people use R, whether they can process this. Anyone has experience with R?
The amount will be 2 by 31.
2 by 31. What that mean? 2000? No. The size of the matrix? The larger than this or smaller? Yeah. Okay, so in this case, I'm going to try to kill this process because this matrix is overwhelmingly large. I'm talking about this. Okay. The zipped matrix file has 337 megabytes, and after unzip, I don't know how many cells in there.
Yeah, Alex, what's the error that
Alex talking about it's a memory issue.
It's also like 496 gigs.
Okay. Okay, so it's confirmed I have to kill this, otherwise I will run into the same problem. So kill this process. What I use is in the command window, use Control-C, and sometimes you can stop the procedure, but sometimes you don't. And then you clean all. So I gave up this, and if you don't have time to work out for the next 2 papers, I think we just leave them like that. Now I'm going to show you if you have a local file, what you can do. So you run Cell Ranger, and Cell Ranger will give you an output, something like this. I have an example here. Cell Ranger will have a folder that's in my download file. So I prepared this as an example. In Cell Ranger, you will see, okay, this filtered feature BC matrix, that's where the 3 files is located. So that's from your own data. So if you have this, what you can do is to select 10x genomics alt folder. Okay, so that folder is called alt folder. So all these options above this line are local file selections. If you have a local MTX file or you have a TXT based, rather than you give a link from the geo database link, then you can use this local options. Here I will select this folder. You don't have this example, just watch, and just in case, just as though you have that. I will select the folder I'm just talking about. And I select this folder. And then the program will find the 3 files in that folder and start to read information in. By the way, those files are zipped. They end up with the GZ, and so we unzip them and read that in, and then unzip them, zip them back. So you still have exactly the same files in the folder, but the information has been already extracted. In this case, after you read this data files in, the metadata will tell you when this object is created and also which folder you import this data. I don't know if it's a good idea, but your folder name will be released somehow. Okay. Yeah.
The bit paper, it's like a TAR file. How do you open that?
TAR, let me see. So we have a question. This is the last paper. Okay. This file, okay. This link. So the last piece of this example. Oh, so we have a lot of individual samples. They put 3,000 of them. And then we have, oh, you mean this one, TAR file? Oh, this, I don't know. Okay, so that's the one from local. That's raw data. Okay, in this case, we have to download and unzip and see what's inside.
Okay.
Yeah, so we have to process this locally because they didn't give you any information about what's in there. It could be MTX, could be text-based, so we don't know. Right? I can just download and unzip it and see what's in there. So in this case, I will treat this file locally and see, depends on the cell types, I can get that in. So you see at a different scenario, you have to treat this condition differently and then to find a way to get data in. And sometimes you really, the author just make mistakes. They didn't give you the gene name, or maybe they have a wrong format of data. So in some cases, you just cannot get the data itself. Okay. We have to accept this. Okay. So for this particular example, I download the raw data and see what they put in. You know what I think? This is called a super series, and so under this they have several different sub-records. Maybe one of these should be the one we need. So I downloaded this data. Let me see if I can open this. Inside here, we have the mixture of, I think, different sub-project data. For example, if you are interested in one of this, you should be able to, yeah, so I will, for example, I will do I will come here and to do the local MTX. And then read one of these. So in my download folder, I should have, well, yeah, I should, for example, click this. I don't know if I need to unzip it first. Features, yes. Yeah, so I need to find a way to unzip my file first, then read that in. So in this case, I will see, okay, I'm only interested in this sample, so I will delete those and unzip them, extract here. Now delete those zipped file. And now I should be able to read that in using this. Okay. By the way, this is the local 10x genomics alt folder. So I read that folder in, and that's the data. Okay, it's time for a break. 5 minutes and come back at 11. see. Still online. Okay, so I want to show the new feature like a view metadata. So this indicate, okay, I can remember now where I get this data from. Okay. And for this one, view the metadata. Okay, I know it's from this folder. Okay. So when you have any feedback, I should be able to respond very quickly. So last time you say, okay, there's no metadata, I can put that in. So when you use it, let me know when you have any problems. So a lot of those experiment comments are from the PIs or from students. Say, some of them say, I want to see a marker genes, but I want to separate cells by express this marker genes and not express that marker genes. So I can build that function in, and it will be helpful for other users. Okay, so the last thing for the data importation I will show you is how can you import multiple data at the same time. I will use example to do this. Okay, in this session number, so that's example. So let's see. I will go back to geo session. So you see that example data is from this study. It's a mouse study for beta cell, and they have several samples at a different age. So week 8 and week 14 and week 16. For example, if you want to have two data sets to be imported side by side, so I'm going to use this as example for week 8 mouse and this as a week 14 mouse. So I'm going to import two data sets at the same time. So you have two batch of data. And you can do multiple as long as you know the different batch, what that means. But in this case, I'm going to use this end up with 8547 and 8548 as an example. So when I select a session number here, I can put multiple numbers here. And the 47 is the younger mouse. And then I copy it and put a comma here and give this the next one. So I'm going to import two samples at the same time and merge them. So I will download the data and merge it into one data set. So you have to make sure you have a clear separation of your samples and each under that ID their data in there. Otherwise, if authors provide a combined data set, you have to process them differently. But here I give you an example. If they provide individual data for different samples, you can use this to get the data together, merge. And you will see two batch of data, one colored in blue, another colored in red. So you can separate them visually. On the screen, it shows my first sample has been processed. Now the program is doing the second sample. Then hopefully the merging function should work properly. And I should get data in. If not, let me try next time. Okay, let's see. Are you trying this at the same time now? No? Okay. Do you see this? And are you going to merge? And so if you decide to do this, everything automatically, the merge and then do the clustering. I will do this manually. So right now I will skip this step. Okay. So let me see what's the metadata tells me. And the first sample is from there and the second sample. The first thing I can do is to label the cells by batch ID. And you will see blue and red cells. To label the cell, you always use this label the cluster. So now I'm going to do a quick filtering by the QC index. So after QC, what's the next step? Do you remember? Yeah, so I'm going to do the embedding using Disney. So the button is this one here. And I should see some kind of separation of two batch of data if they are different. If the two batch of data are highly similar, and they should, or the cells will be mixed together, you cannot separate them. Here we go, and I see the separation of the majority of the cells into two different clusters, multiple clusters, but you see clearly different between this young and old. I mean, it's not a young and old, it's different weeks of the age. So with this, I'm going to annotate it. First, I'm going to cluster them into a lot of small clusters, right? And put a big numbers, and then annotate the cell types manually and mouse. Okay, it's done. So we merge those cells have the same name, and there are 12 distinct group of the cell types. Okay, that's our annotation. Now, because you have two different batches, so we can use this button to see I want to view multiple groups. I want to separate the cell type and the batch ID. Now you have this view, and on your left side is cell type you just annotated. On your right side, you have your batch, the first group and the second group. So you can view them side by side. For example, I see the cluster of here, this one, this group. This only batch one, so data set. And the cell type is the beta cell. And then this group also is beta cell, but that's from the another batch because the color differently. Okay, regarding this group, so you have doctor cell, hepatitis, and this group, blood cells. So it looks like both samples have some blood cells because if you highlight this part, you see, right? You can rotate this, so and also if you want to label them, just label everything. Okay. Any questions regarding this function?
Could you do this with local files?
You can, yeah. I will show you later how. Yeah. Okay. Next question. You normally people. So here is the multi grouping view, and another question is you want to have statistics. Say you want to see in different batches what's the distribution, what's the numbers of different cell types. So you can click this by looking at a bar chart like this. So this tells me first is alpha cell has a lot in the sample. We have more alpha cells than any other cells because the total number of cells have a lot. And in batch 1 and 2, similarly, you have same proportion of this same type of cells. Well, the beta cell, we have very few, but the majority of beta cells is from a batch 2. You see here is the fraction, and that's total number, the real number. Oh, that's a B cell, not a beta cell. We have endocrine cells as a second, and then batch 1 have more of this type of cells than batch 2. So you have this annotation and also compare different composition of cells between batches. we can have a more than 2 groups if you have a multiple samples. You see, okay, I have a week 8, week 14, week 16. We can have that because we do have the function to import multiple samples. All right, I think we are okay with this, playing with this toolbox. We are going to move to lecture slides. Okay, any other questions? No. Yeah, so next time we can find the time to show how can you have multiple data locally, and you can merge different data. And basically, you have the function to merge your separated objects, because each of this you can save as a save in the workspace. So you have multiple, then you can merge them. Any students online have questions? No. Now I'm going to go over the slides, and once we have any computational related subject, the topics, we can try them out in the MATLAB. So the first thing is from Tuesday. So if you have a matrix like this, how are you going to do the normalization, library size normalization for this data? Let's get example data in there. So what you can do is to use this example data. Okay, we can load this because we need this X matrix. So you load this data and then export. So here, this button, you can export the data to workspace. We don't need SE, we need big X, right? This is the matrix, and you need a list of gene name. So once you export these 2 variables, you will be able to see them in the workspace. So I just click this, you see here. Let me know once you finish this, so we have some data to play with. Then you can shut this down because we already have data. Close this. Now, first I view the list of genes. That's genes, and that's the matrix X. Okay, with this, tell me what you can do with this right now. Anyone need any help to, yeah.
Do you normalize the total expression per cell?
Yeah, we are going to do that, yes. So let me know if you need more time to get this X matrix. So you have something to play with. Okay, you are okay. Okay. Yeah. So what I'm going to do first, I will do a sum x. Okay. This is by column. So you know this will be a number. See, let's see. Okay. I mean, size. Okay. And you can put a dimension indication. Say, okay, I want to do this by dimension 1, so that will do the column sum. If you do 2, that will be row by row, sum by row. Okay. So if you don't do anything, then it will be default by 1. So this is the library size. So you will have number is a vector of 2,509 because you have 2,509 cells. So that's number. The first cell you have 2,000. So the total number of UMI. This second is this. So it looks like the second cell has larger library size than the first one. Okay, how are you going to see the distribution of this library size? So the question is, if you have this list of cell number, you have this vector, and what you want to do first with it? Histogram, yeah, histogram is really good option. So histogram of the size, this library size. So the first thing you can do is very easily. You see this beautiful histogram curve, and you see we had a cutoff for the data. So there's no data smaller than 500. So the smallest, the minimum of library size is 1,000. So we had a cutoff 1,000. Well, we don't have a cutoff for the largest value. So the largest library size in this case is over 14,000. So this cell maybe only have 1 or 2 cells in here. They definitely outliers. Okay. Okay, we have some impression of this. So we know on average, we expect to have about 2,000 of cell, library size 2,000. So that's a reasonable number. So the quality of data is fine. And sooner or later, we may want to get rid of those cells which have an extremely large library size. So let's see, can you figure out, see visually, can we set up a cutoff? Are we going to use 7,000 as a cutoff? So any library size greater than 7,000, we will delete those cells, remove those cells, and we're not using it.
My histogram isn't like popping up when I'm doing the function.
Pop up, yeah, it's maybe it's hiding behind it. Oh, what do you mean by pop up?
Just when I'm running the function on that.
Not showing in here. It's behind the window. And yeah. Yeah, so histogram is one way, and you can also use what else so to see this distribution? Box plot, maybe? I don't know. Eric, you are using this. So box plot could be a one. So you see something like this, and you say, okay. but the histogram should be the first option, because this is not clear. And I don't know if you can use CDF plot to see this. Okay, so those are some options. Histogram definitely the one very similar to. I don't know if we have a PDF plot. I'm not so sure. We don't. Histogram is PDF plot. Well, so we know we have a cutoff of 7,000. So we want to get rid of the cells with more than 7,000. So what should we do? And is to do size greater than 7,000. So I'm doing an operation to compare each element of this to 7,000, see whether it's greater than that. So that will be indicator. Okay, have an indicator of this. Maybe just index. So this index is logic. So when you have 0, that means false. When you have 1, that means true. And you should see a lot of zeros because the majority of this library size is smaller than 7,000. Now we are going to remove. If you do this index, go to nothing. So this operation will using that index. Once the index is true, that column will be removed. Okay. Now, before remove, I have 2,509 cells, and after remove, should have I removed 9 cells. Okay, now I have 2,500. Okay, I will compute the library size again with the new matrix, and then do the histogram again. Now, the range is from 1,000 to 7,000 now, because I have a cutoff and get rid of 9 cells. Yeah, so I just showed you how to do this sum by column and the sum by rows. If you have a 2 in there to indicate the different dimension, and then so you have the library size now. So you can do this, which I highlighted to well, is this divided by the library size? Well, is this correct? I'm not so sure whether this is a. Let me check my norm library size. So that's my source code where I do the library size normalization. Yeah. So I view yeah, so you just divide by the library size using the matrix. So that then you get a normalized matrix. So I have my x, I will divide by, so x normalized library size. Okay, I'm done with my normalization. So you can take a look of this new matrix. So now if you compute the library size for the normalized, you should have 1 for all the cell. Okay. So it's done. The normalization is done for the X matrix. You got Xn, which is normalized matrix. Any questions for this part? Yeah, so you see the value is really small in this case. So that's why we want to have a constant to scale it up a little bit. But that's not essential. Okay, so move to next section. So we have normalization. Yeah. So now let's see if I can generate a data. I'm going to generate a random data. So here I generated 5,000 random numbers. So if you don't know its distribution, the histogram will be the option and to take a look. And then where is it? So you see in this case, the number generated is by uniformly distributed from between 1 and 0. So you see this flat line and a straight line here. If you do use another random generator function, so in this case, you have normal distributed data. Okay. So we are doing this generative model experiment using different generative model. And one is a uniformly distributed number, and this normal distributed. And can't remember the Poisson distribution. There are other random number generators. Yeah, we have to use statistic toolbox in order to find specific distribution random number generators. Okay. I don't have that topic from my head. Maybe the negative binomial, we do have this, so I'm going to try that. Okay. Negative binomial D. If you have 3 and 0.01, then you want to generate, say, 5,000 data. Okay, so now this number is for these two parameters, and you generate 5,000 random number based on this distribution. And this histogram for it. Okay, so that's the negative binomial distribution. Okay, I hope you can explain what that means here. So if I have value in this bin 16, and the small, the probability is really small, but the value is between 0 and 30. So some maybe I would say, okay, 15. If you have a number 15, you will be in this bin. So what that means, the 15? 15 in this case means if you have a trial which have a successful rate 1%, and you want to get 3 success events, how many you have to try before you can get that 3? So if you say, okay, I tried 15 times, then I get 3 success. The likelihood to have only 15 trials is really low. So you have this low value here. But on average, you need to try over 200 times, slightly more, in order to get this 3 times success. Okay, we had histogram, we have a PDF, CDF, box plot mentioned. What else we should? Poisson distribution. So in the class, we already showed this, right? You can compute the probability for given parameters. Actually, not the parameters. Poisson only have one parameter. That's how the probability is computed. Okay, CV. So we can give this a try. Okay, I'm going to load my X matrix again. The way I'm going to do it, to load my example data and export the X matrix. So I have something to use. Okay, so I will do the library size first. I will remove some cells. So gene, I didn't touch genes, so the same number of genes, so I don't need to change the gene list. So I just get rid of those. And then I will do the normalization. I will use the same variable name, okay? Just overwrite that. Yeah, I need to compute the size 2 again, size. Okay. So I'm using the same variable name from input and output. I save some variable name instead of xn. I just replace x itself. So now I can do a little bit scaling. So 10,000 times. That's my normalized x. Okay, I'm going to compute the average expression of gene. Now I'm going to compute this for gene. So I have my average compute for x, but by gene. So that's my mean. Okay, so I have a normalized expression matrix. Then I will compute for each gene what's the average expression. So this M is average of for gene. So you have 3,850 genes. So this vector is that. Okay. Any questions so far for those functions? Is what we are doing here? So in order to compute CV is standard deviation divided by mean. So I have a mean right now, so I need a standard deviation. So let's just call S. Okay, that's my standard deviation. So my CV is S divided by mean. So I just follow the definition and what is the coefficient of variance, and then standard deviation divided by mean. So everything is gene wise. So in that case, the column, no, row based. So I have the 3,800 for the mean, for the standard deviation, and then the ratio between them is my CV. Okay, so each gene has its CV, has its mean. So now I'm going to plot CV against the mean. So that's a scatter plot. So you have mean in the x-axis and y-axis. So that's your scatter plot between mean and the CV for genes, right? We are going to take a log instead of using the mean itself. So let's do a log plot. Now you start to see after the transformation, and then you almost see a straight line here. And some genes not follow that. They have a more than expected. So that's called over dispersion. You have more than expected standard deviation. That means you have a most dispersed values in its expression across cells. But because the whole population, if you consider all the genes, so each point is a gene, you're supposed to have this straight line here. Every gene should be on this straight line if they are not over dispersed. You may have question whether we should use nature log or time-based log. It doesn't matter. So you will have a similar result. Okay. The reason you should expect a straight line negative correlated is explained by the writing here. So because we assume Poisson distribution of gene expression in cell populations. That's why you expect to have this relationship because of this assumed model. And the model seems correct for majority of genes. So that's why we see this straight line here. Okay, so your job right now is to put that vector in. So you have this example data with 6 numbers, and use MATLAB and to see how can you do this and get this value. Let me see, I can zoom this a little bit. Okay, your job right now is in MATLAB, put in this vector 17, 15, 23, and then see if you can compute in the same way to get this variance. And you can certainly use function and the VAR to get the variance by one step. But I want you to try out the vector minus its mean and then squared. Sum it and divide by the n minus 1.
Question.
Yeah.
So after you clean up the data and normalize it, do you have to save it as a new file to reopen it in the toolbox?
Yes. So you don't have to, because this process is very quick. It's so easy. So you can have a raw data, and anytime you want to normalize, just take 1 second. So we don't save it unless you process data take several hours. You want to save your temporary data for later use. Yeah, but library size normalization just nothing, not cost anything. So we don't save the intermediate step of those. Yeah. Good question. Yeah, I think that's a good example. This awesome looking mathematical equation, and can be illustrated with this table, and in MATLAB it's just one line of code. And let me show you how. My data is 17, 15, 23, 7, 9, 13. Okay. To centralize the data, you just minus its mean. Okay. So that's the centralized data, and that's this column. Then you can have a sum of this squared. This should be, yeah, 166. And then you want to divide by its number. So length of D minus 1. So this will give you the variance. If you don't want to do this, this variance function should give you the same answer. Okay. Okay. That's all for this week.
I had a quick question, if I could.
Yeah. I think yes.
I think it was the previous example for the norms, you used empty brackets as one of the inputs and one of the commands, and I was wondering what that meant.
Oh, when I do the standard deviation.
Yes, I think it was.
Yeah, when I do standard deviation, TDS, is this one here?
Yeah, I was wondering what those empty brackets, what that meant on the input.
Oh, they are the second position supposed to be an index to tell you whether you want to use n or n minus 1 to compute the variance. And the third variable is the dimension. So that's why I leave this blank to let the function itself to decide.
Okay, would it
Yeah.
still work if you put a space there? I wonder.
If I put 1, it will have the same answer.
Okay.
So the answer of this S1 will be the same as if I put nothing. So is equal S1 S0. This should be the same. Well, let's see. Oh, so so if I put a 0 here, this should be the same. I believe 0 and S and empty should be the same. If you put a 1, then so you should look at the documentation here. And the second, W here means their explanations of why you change the W. Okay.
Okay.
Yeah. So the third variable is a dimension indicator. So just do a
Okay.
DOC and then the function name.
Yeah, I
You
just
have a
looked it up on
documentation.
Google, actually.
Yeah.
And I got the
So,
same
so if you
thing.
don't put anything for the W or for the empty, it will by default.
Okay, then it's by
equal
default
to
0. Okay.
Yeah.
So is the empty bracket the same thing if I put a space there? Just a blank
I
space?
don't think so. The space shouldn't work because the space, if you put a space, nothing here.
Yeah.
No, you need something here.
Okay. Okay.
Yeah, you need a space should be. Okay. Good question, and see you next Tuesday. Thank you.